Jen, thank you very much. And I think that you can hear this. As you have probably figured out from all of that, I've been interested in keratoconus for literally decades. I think it's a fascinating disorder, and I can't tell you how far we've come in biologically. From a scientific point of view, it's fascinating. But I can't tell you how far we've come in what we've learned in the last 2 decades. So what I'm going to do today is, first of all, I'm going to review the structure of the normal cornea. And then I'm going to discuss the pathology of keratoconus corneas, focusing on 3 things, because these are the 3 things that are important for keratoconus. There's corneal thinning, there's anterior corneal instability, and there's high levels of oxidative damage. Then I'm going to talk about some of the molecular mechanisms, and we're not going to get into equations or anything too complicated, but just the overall concepts. And then I'm going to discuss some of the genetics, because people say, gee, I have it, but nobody else has it, or it runs in my family. So we're going to talk about that. And then finally, I'm going to summarize the research from a very large international meeting that was just held in Seattle, actually 2 weeks ago. So this is a diagram of the normal cornea, and I think most of you know the cornea is the clear part of the eye, and this is a schematic of a cross section. So if you take a piece of cornea and cut it in cross section, the cornea actually has 5 layers. The first layer is the epithelium, which is a layer of cells, then Bowman's layer, which is this matrix that the epithelium sits on, and then the stroma, which is the bulk of the cornea, and then a specialized membrane called Descemet's membrane, and then a single layer of cells called endothelium on the back of the cornea. This is just what a cornea looks like, that it's been sectioned and stained, and you can see here's the epithelium, you can see the bulk of it is the stroma, and then the endothelium. Now, in the cornea, 70% of the total dry mass of the cornea is collagen. And I think most of us are familiar with collagen. There are 2 kinds of collagen, the fibrillar collagen, such as the kind that you find in tendons and skin, and then there's this non-fibrillar collagen. And the non-fibrillar collagen acts as packing material. So the fibrillar collagen makes a structure or a scaffold, and then the non-fibrillar collagen sort of tacks it together. And then with that scaffold, you need packing material. And that packing material are called proteoglycans, glycoproteins, and different kinds of other proteins. The secret of the cornea is that when you put all of this together, it attaches to each other. It doesn't slip. You've got connections and anchoring fibrils and things that really keep it firm. And this is just an example of this diagram is a scanning electron microscope. It's blown up 1,000 X magnifications. But the point of this is that the cornea is actually layers that are interwoven. And you can see this is one of the layers here, a lamellae, and you can see it's bifurcating here, that's another lamellae. And here you can see this one goes above it, and that one goes below this other lamellae. And the point is, it's sort of like a basket. It's all woven together, and that's part of what gives it its stability. Now, in terms of keratoconus, it's stated that the incidence of keratoconus is about 1 in 2,000 individuals. It usually appears in teens and 20s, but as Dr. Bryan said, it doesn't have to. Sometimes people are younger, sometimes they're older. It's usually unilateral in only 10% of the cases. Most of the times people have it in both eyes. And there's a family history in about 6 to 10% of the patients have this family history. Now, when people do studies on keratoconus, this is just the basic study design. They obtain the corneas from transplants. They also get normal corneas. Is it from here? Okay. normal corneas. And once you get those corneas, then to analyze them, some people culture the cells, other people isolate the DNA, RNA, and proteins, and then other people get the corneas and then look at them with various kinds of microscopes. So, as I mentioned earlier, there are 3 pathological features of keratoconus. And the first is corneal thinning, second, anterior stromal instability, and the third is oxidative damage. And what we're going to show that all of these features are related to each other. Now, why does the cornea thin? What's different about the keratoconus corneas? People have been doing studies and they found that the keratoconus corneas have increased levels of what are called degradative enzymes. These are enzymes that are present normally in our body, but they're inactive. But with keratoconus patients, they have a lot more activity, and therefore they degrade and chew up the collagens and the proteoglycans that are in the cornea. The second thing is normally there's a balance between these degradative enzymes and inhibitors. But what happens with keratoconus patients is that the inhibitor levels are lower. So you have lower inhibitors, and therefore you have higher levels of enzyme activity. Now, people have been studying this since the 90s. There was a lot of effort in the 90s to identify these different enzymes. And you can see it wasn't just one enzyme that was more activated. There's a whole host of enzymes. There's lysosomal enzymes, this category, cathepsins, and then these categories are the ones that digest the collagen. The matrix metalloproteinases digest collagens. They also showed in those years, the 90s, that they have decreased levels, not just of one inhibitor, but of multiple inhibitors. The tissue inhibitor of metalloproteinase and this proteinase inhibitor and the macroglobulin, they all inhibit different pathways. So then this is like a double whammy, because it's not just one single pathway that is changed, and therefore we can go over after that one inhibitor or that one enzyme. There are multiple enzymes that are increased and multiple inhibitors that are decreased. So the consequence is this. Normally, you have collagen and matrix that's produced, and it's in balance with the normal degradative enzymes. It's normal to have degradative enzymes. If you didn't, you would just keep having so much matrix, and there are diseases where you don't have enough degradative enzymes, and it's not good. But with keratoconus, you have increased activity of these degradative enzymes, and therefore you have thinning of the matrix. Now what the result of that is here's our schematic again. Here's our epithelium here. Here's our Bowman's layer. Here's the stroma, and that stroma is packed with all the different kinds of collagens. It has proteoglycans, it has glycosaminoglycans and different proteins. You have Descemet's membrane and the endothelium. So there's a normal thickness to a cornea. But with all those degradative enzymes, then it chews up all of that matrix in the stroma. So you get a lot of thinning. And in addition, in the Bowman's layer right here, the enzymes degrade a lot of the Bowman's layer. The second feature is the anterior corneal instability. And I'm going to present 3 different studies that were done using different approaches, all of which came to the same conclusion, that keratoconus is mainly a problem with the anterior part of the cornea and not really so much involving the posterior part of the cornea. The first study was published in 2009 by Keith Meek, who's in England, and he had noticed that yes, you have the increased degradative enzymes, and it weakens the matrix, and this causes stress and tears in the stromal layers, especially around the apex, the base of and the base of the cone. So what they did is they used this very special instrument, this wide-angle X-ray instrument that measures scattering patterns of the fibrils. And so they can actually look at how the collagen is spaced. And this is the normal cornea, and I think you can see that the patterns here, they're very uniform for their patterns, as opposed to the keratoconus cornea, that you can see there's a lot of irregularity with the fibril arrangement. The second work that was done was by Dr. Bermanson, and what they took, it was a really tough project, because what they did is they took normal corneas and keratoconus corneas, and they did serial sections, and that means one section after another section after another. So they made ribbons of sections of corneas, and they looked at hundreds and hundreds of corneas, because they wanted to trace the fibrils, because we didn't know if they were stopping, fragmenting, you know, what exactly was happening. So he worked on this for a number of years, and what he found is that there was the thinning of the Bowman's layer, but he also found a lot of fragmentation of the stromal fibers, excuse me, and loss of that interweaving that we saw earlier. Excuse me. And this just shows, this is a normal cornea. Now this is a transmission electron micrograph. So you've taken this cell and you've blown it up to the size of, you know, this size. So this is just one part of one cell. And this is the Bowman's layer. But in a normal cornea, you can see how thick that is. But in a keratoconus cornea, it's much, much thinner than in the normal. The other thing that's very important is as these layers break down, then the epithelial cells, the cells that are supposed to be separated from the main part of the stroma, they start to invade in. And they're areas that they again cause weakening. So the third study was published in 2007, and they used a confocal microscope. And this is a very, very fancy microscope that it doesn't use light to look at the images. It uses a laser, this femtosecond laser. And it generates these signals. And with these signals, you can actually look at the collagen in the corneas. And so he also looked at normal and keratoconus corneas, and then reconstructed three-dimensional maps of these corneas. And this is what a normal cornea looks like. Here's, again, your epithelium, your Bowman's layer, your stroma, Descemet's membrane, and endothelium. And what they found are these, what they called anchoring fibrils. They go from the anterior part of the cornea to the epithelium. And here you can see them in the normal cornea. And they're literally anchoring the epithelium and the Bowman's and the anterior stroma. So it's really providing stability. Well, what does a keratoconus cornea look like? Keratoconus cornea barely has any of these. They really, really are absent. And so again, if you don't have all of these little anchoring fibrils, you're going to get instability. So here's your keratoconus cornea. You've got thinning of the Bowman's layer. You've got fewer anchoring fibrils. You have less matrix. And so when you have the normal pressure that you have in the eye, it's going to bulge it out. And so that is part of the mechanism by which you get the bulging. Oops. The third aspect that I'm going to talk about has to do with oxidative damage. And the first thing I'm going to do is give you some definitions. Reactive oxygen species and reactive nitrogen species we refer to as NOS or NRS. Oxidative stress is when you have an altered balance between how much ROS you're producing and how much you remove. ROS is the same thing as free radicals. Hydrogen peroxides, superoxides, things like that, those are free radicals. Those fall under the category of the reactive oxygen species. If you have oxidative stress, then instead of getting rid of those free radicals, they accumulate and they cause damage to the tissue. When the tissue is damaged, then you have decreased cell function and cell death. Now this is, I want you to memorize this because we're going to have a test afterwards, and you can't leave until you pass it. No, this is just basically a series of biochemical reactions by which free radicals are formed, or reactive oxygen species are formed, okay? And so the reason we have it is that everything that's in yellow has been shown to be elevated in Keratoconus corneus. So normally you'd have this, all of these are reactive oxygen, nitrogen species. Here's hydrogen peroxide, but they're all elevated in Keratoconus corneus. Everything that has a little asterisk by it, all these little enzymes, these are various enzymes, catalases, superoxide dismutase, those are all enzymes. Those are all altered in Keratoconus corneus. So you can see, if you just take a Keratoconus cornea and you look at it, everything's off balance, it's off kilter. The enzymes aren't working right, you have too much reactive oxygen species, and that can be a problem. Now, this is just to simplify it, is, okay, here we have the reactive, the ROS and RNS. Where do they come from? Just ultraviolet light, sunlight, as Dr. Bryan was saying. Rubbing the eyes too hard can irritate the eyes. There's an organelle, or there's a little tiny structure inside a cell called mitochondria. They normally produce reactive oxygen species. It's normal to have these. Everybody has them. But fortunately, we all have antioxidant enzymes that get rid of and keep everything in balance. But with keratoconus, as the previous slide showed, we don't have all those antioxidant enzymes. So what happens is that you have an accumulation of the ROS, and they are funneled through different pathways. One pathway is called lipid peroxidation, and that forms aldehydes. Well, we know aldehydes aren't good. And the other is through the nitric oxide pathway, and those are formed peroxynitrites. Both of these cause oxidative damage. When you have oxidative damage, it feeds back and increases the degradative enzymes. So it's sort of like a cycle that is occurring. Oh. So how do we know that we have elevated peroxynitrites? There was a study done in 2002, and what this group did, actually it was our group, what we did is we took normal corneas and keratoconus corneas, and then we took corneas that had other diseases, and we analyzed for the amount of peroxynitrites. And you can see in the epithelium, the stroma, and the endothelium, the keratoconus corneas had much, much higher levels of the peroxynitrite than the normals, and even higher levels than corneas from other diseases. Now, I also had mentioned that the mitochondria generate some of the ROS. So what are mitochondria? So now we're going back to high school biology, okay? So here's a cell. Here we have the nucleus of the cell, and what mitochondria do is they provide energy for the cell. They're like the batteries for the cell. So every cell has mitochondria. We absolutely need them. Now, mitochondria are really special and unique because in a cell, in the nucleus, everybody knows we have DNA. You get half your DNA from your mother, half the DNA from your father. Mitochondria have their own DNA, and you only get it from your mother. Your dad doesn't give you any of the mitochondrial DNA. So that's what makes it so good to analyze. And the other thing is unlike the nuclear DNA, which is the double helix that we all learned about, the mitochondrial DNA are little tiny round circles that are found within the mitochondria. So what we did for this study, we extracted the DNA from normal in Keratoconus corneas, and we used various techniques. And what we found is that the mitochondrial DNA had a lot more damage than the normal DNA. So if you look at the average here, this is keratoconus. The average was 7.4. It's almost double what the number of defects that were found in normal. So why is that even important? It's important because the defects, if you have defects in mitochondrial DNA, that can lead to increased ROS production. If you have increased ROS production, you're going to get decreased energy levels, because that's what the mitochondria do. The function of the mitochondria is to give you energy. If you don't have enough energy, the cells stop functioning. They just give up. They can't. They require a certain amount of energy, and once they really give up, then they die. So again, this is just going back. It looks complicated, but it's just I'm trying to get the concept there, that you have all of these pathways involved with the elevated reactive oxygen species, with the abnormal antioxidant enzymes. They can cause damage to the DNA. Here our mitochondria is damaged, and then this feeds back, so you get more ROS, more oxidative stress, and it's just a one big circle or a cycle that's happening. So when we ask the question, are these events related? The question is yes, because if you have oxidative damage, it's going to cause thinning because of the increased enzyme activity. The increased enzyme activity is going to digest the matrix of the cornea, and that makes the cornea unstable. So the cells that are in the cornea, they don't like it. They become stressed, and then they produce more oxidative damage. Now, I'm going to switch gears a little bit, because there was a very interesting paper from last year that was published, because everybody sort of thinks, okay, all of this is going on in the cornea. So this group wanted to know, well, how about in the tears? Are the tears, can you follow keratoconus patients, or can you identify markers in tears? So what they did is they collected the tears and the epithelium from normal patients and keratoconus patients. And they found, when they analyzed them, that the tears had higher levels of the degradative enzymes and inflammatory proteins. These should not be at high levels. And in the cells themselves, they had high levels of the degradative enzymes and even a greater number of inflammatory proteins. So what they said is, okay, there's oxidative damage, stress going on. What if we treat the patients for 6 months with an anti-inflammatory product? And that's called cyclosporin. It's an eye drop that is can be used in ophthalmology. And they treated it for 6 months, and they found if they did that it reduced the levels of some of these degradative enzymes, and there was a local reduction in the curvature of the cornea. Now, they are starting clinical trials. When people start clinical trials, they're assigned certain numbers, and so they're working on that. Here's the clinical trial number. I don't know where they are with the clinical trial. I don't know how long it's going to be going, but this paper was just published, so it may still be ongoing. So that's sort of where the background of keratoconus and why it happens. So what I'm going to do now is I'm going to switch to the genetics of keratoconus. And people have been looking at the genetics of keratoconus for 20, 25 years. Now, again, high school biology. In every cell, you have DNA. The DNA codes for the RNA. The RNA codes for what are called amino acids. When you string the amino acids together, that forms a protein, and that protein gives you a function. So that's, now what we're going to do is look at the DNA, because if the DNA is bad, then this is all going to get messed up and the function's not going to be good. Okay, so how do we know that genetics are even involved in keratoconus? People have shown that there's increased keratoconus in identical twins compared to the general population. So if one twin has it, if they're identical twins, the second twin is going to also have it. Second of all, as I mentioned before, 6 to 10% of keratoconus patients have one or more family members with keratoconus. Now, this is a very, very good article that just came out in 2014, and what this group did is they took all the studies that had been done all over the world on keratoconus and genetics, and they put it together as a big review. And what this table shows are, these are different chromosomes, and we're just doing general stuff. They're different chromosomes, and these studies were done from studies from Australia, from Italy, from Saudi Arabia, Hispanic and Caucasian, Italian, Finland. The take-home message of this slide is that 16 chromosomes have been associated with keratoconus. It's not one chromosome that everybody has and then you get keratoconus. It's 16 chromosomes that have been identified. What this slide does is, rather than look at the chromosome, they've looked at specific genes. And again, the take-home message, and this time they've looked at people from Ecuador, Korea, China, Japan, Europe. At least 20 of the genes have been associated with keratoconus. And finally, the VSX1 was the very first gene associated with keratoconus. So you think, okay, there's one gene associated, it's going to be very straightforward. No, no such luck. At least 8 different variants of that gene have been associated with keratoconus. Not only that, keratoconus has been associated, many people just have it in their eye, they don't have any other systemic problems. But there are many disorders, such as Down syndrome, that have people with Down syndrome have a very high incidence of keratoconus. Some studies say 10 to 300-fold higher, and along with these other syndromes. In addition, there's some very rare syndromes that you hardly ever see. I haven't seen 80% of those syndromes I've never seen. So they're very, very rare. But in the literature, you can find that those people oftentimes will have keratoconus in addition to this syndrome. And that's also true for the Marfan syndrome and the Ehlers-Danlos syndrome that we were talking about. Yeah, right there. The take-home message for all of this is that the genetic cause of keratoconus is very heterogeneous. There's not one single gene that is associated with keratoconus. And the keratoconus, it's a very, very complex disorder. There are multiple genes that are involved. Now, there are modifiable risk factors because we know that oxidative damage and oxidative stress plays a role of that whole cycle. So there are the risk factors that we can modify are the exposure to light, the eye rubbing, poorly fit contact lenses, and chronic irritation. But I've always felt that if we were going to treat keratoconus in a non-surgical way, that you really have to identify and intervene in the common pathways, and you can't target one gene that you're going to replace in the individuals. Now, this is my view of the cornea. So with the normal cornea, we normally have stress, we go outside, we rub our eyes, we do various things. It produces some ROS, but we have a lot of enzymes that just come in and get rid of that ROS, so you don't have damage to your mitochondrial DNA and you have normal functioning cells. But in keratoconus, what happens is those patients can be exposed to the exact same stress, but they have abnormal enzymes, and so those abnormal enzymes can't remove the ROS. So there's an accumulation of the ROS. When that happens, you get some mitochondrial DNA damage, it causes some damage to the cells, more ROS is produced, they have abnormal cell function, and we know that the ROS can activate the degradative enzymes, and that's going to lead to corneal thinning. So what can we do about it? We've gone over a lot of things. You know, we've got a pretty good idea of the kinds of things that are going on, but is there anything that we can do? Well, there are two categories of risk factors. There's the non-modifiable risk factors, things we can't do anything about. Our family history, our age, our gender, our genetics. But there are modifiable risk factors, such as smoking, obesity, eating habits, rubbing eyes. These kinds of things generate oxidative stress. So the goal, what I've always recommended for patients, is to really try and control some of the levels of the oxidative stress. And for patients, very simple things like use really good protective. It's, as Dr. Bryan was saying, check whether or not your car is letting in the UV light. Get some good sunglasses. If you have contact lenses, do they have UV protection? A lot of keratoconus patients have what's called atopy or allergies. Their eyes itch all the time, and so they rub them. Well, we want to avoid vigorous eye rubbing because, again, that can stress the cells. Some people, they'll have contact lenses. They know the contact lenses don't fit. They can tell, they can barely stand them. If those contact lenses sit on there, they're going to rub and irritate, and it's just going to cause more problems, and that's going to cause inflammation. So what I've always recommended for patients is that you use the protective UV protection for your glasses and your contacts. Get properly fit contacts. Nowadays with the scleral lenses and some of these other lenses, you know, they've made real strides in a lot of the contact lenses, and I'm sure that you have some very good expert contact lens fitters. And I think the number one thing that keratoconus patients learn very early on is you cannot go to Sears and get your contact lenses, or Costco. It doesn't work. You need somebody that knows what they're doing. If your eyes are irritated, if you tend to have allergies, then don't just ignore it and say, okay, well, that's okay, it just irritates a little bit. Be aggressive about either getting allergy medications, non-steroidal anti-inflammatory eye drops, or this cyclosporin drop. You can get these through your physician, your eye care physician, and really try and keep things down, because if you generate the ROS, you don't have the enzymes to get rid of them. So the goal is really to minimize it. And the other thing I've suggested is that try and adopt a healthy lifestyle such as we have for AMD patients. Age-related macular degeneration is the most common cause of vision loss in the elderly population. It's in the back, it's in the retina. It has nothing to do with keratoconus. But the National Eye Institute has put in millions of dollars to study antioxidants, treatments, and lifestyle treatments to minimize the oxidative damage that occurs in AMD patients. Now, they're not going to do that with keratoconus because it's first of all, we can learn from their studies, and there's not as many people. It's a tougher group. It's a small group compared to how many people have AMD. But there's no reason why we can't learn from the 35 studies that they have done. And the number one thing is smoking. Smoking is so toxic to the eyes, and in any case, I would definitely recommend stopping smoking. And in terms of diet, all the green leafy vegetables have a lot of antioxidants. The keratoconus patient may not be making antioxidant enzymes, but you can take them in, and you can substitute. The red meat and the high-fat dairy products they're great once in a while, but you know, it's better not to eat them all the time. The studies have found that fish has high omega-3 fatty acids, and they are very, very good for decreasing oxidative stress. So the AMD studies have recommended fish 2 or 3 times a week, and also any kind of nuts. Now, as I said before, the green leafy vegetables, tomatoes, the vegetables with colors, the kale, the spinach, things like that are really good. Now, let's say I can't stand fish, I'm not going to eat any vegetables or anything green, forget it, fine. Take a supplement, because they've shown that you can get equal effects by taking supplements, vitamin supplements, as by eating the fish. But do one or the other and really try and increase your antioxidants. The thing about a healthy weight is people have done studies and people have found that if people are overweight, that in their blood there's certain factors that are increased expression, the inflammatory factors. So again, it just increases the inflammation that's going on in the body, and that's one reason that sometimes people that have obesity have more medical problems. So then part of this that they've done is they found that studies also are very good to help control your oxidative stress levels, and getting regular eye exams are extremely important for all of this. Briefly, I'm just going to talk about the meeting that was just happened 2 weeks ago. This is a big international meeting that was in Seattle. There were over 5,000 participants and over 6,500 presentations, and over 100 of the presentations were solely on keratoconus. There's so much interest in keratoconus right now. About 24 of the presentations were molecular biology, what factors are increasing, decreasing, how do you control them, how do you regulate them. There were some studies on genetics, but again, nothing saying here's the magic gene that causes keratoconus. Most of the studies were clinical presentations, and the most exciting part has to do with the cross-linking, because everybody, finally there's some way to treat keratoconus with the cross-linking, and there's so many studies that are looking into how to modify it. Instead of using the riboflavin, people have come up with other cross-linkers. Instead of using the UVA light, people are trying to use a laser to get it more focused. So there's a lot of new stuff that's going on. So there's a lot of interest in a lot of things that are on the horizon for the keratoconus patients. Finally, I just want to say, for anybody with keratoconus, if you haven't if you're not aware of the National Keratoconus Foundation, it is a nonprofit organization that was started by somebody that had keratoconus, and the whole reason he started it was he said, I have keratoconus, nobody knows what it is. People look at me and say, well, you know, what are you talking about? You look perfectly fine. And he said, I want people to know and to be able to communicate with other people that have keratoconus. So they have a KC link where people blog back and forth. Oh, I have problems with my contacts, or have you tried this or that? They have newsletters, they have educational seminars. And the other is, again, a nonprofit, Discovery Eye Foundation. This foundation has supported keratoconus research for the last 30 years. And they're both these foundations are very committed to keratoconus and the keratoconus patients. And if you haven't, been in contact with them, the other thing about the National Keratoconus Foundation is they have books. They can send you books and booklets about keratoconus and other things. So they're both really, really worthwhile foundations. And I just want to acknowledge many of the people that I've worked with and that have provided for these studies. And I just want to say thank you very much, and I'm open to any questions.